I'm Emma Holden. I work at the Quadrum, and I'm going to talk a little bit about a project I did a couple of years ago at the Quadrum. So, this is a project I did a couple of years ago. It's a project in collaboration with the Quadrum, Innovate UK, and a drug discovery company called Oplitec. So I don't need to remind this audience that AMR is a massive problem, and increasing drug resistance in bacterial pathogens, one of the ways that we can combat that is by the discovery of novel antibiotics. So Oplitec discovers these novel antibiotics using their computational model, and they had this candidate called OPT200 that was active against gram-negatives. And they came to us with the simple question of, how does it act? So Quadrum is quite well placed to answer this question because of the in-house development of TraDIS-Xpress. So this is a massive transposon mutagenesis screen where we can assay how every single gene, every single locus in the genome responds to growth under any growth condition, whether that be antibiotic stress or anything else. So at the Quadrum, we developed TraDIS-Xpress. It's different from other transposon mutagenesis techniques because of our incorporation of this outwards transcribing promoter in the transposon. So in normal TNZ, we insert the transposon into a gene, it inactivates the gene. You can look at how gene activity versus inactivity affects survival under any stress. Now, in TraDIS-Xpress, when that transposon and that promoter inserts upstream of a gene, you can see how expression changes as well as presence and absence affect survival. And that's really important for assaying essential genes that can't normally be assayed in normal transposon mutagenesis. And essential genes are essential for antibiotic discovery because a lot of the antibiotics we currently use target essential genes. So we use this transposon, this TraDIS-Xpress. That's what our genomes look like. So they're really highly saturated with transposon insertions. So we use this. We used our E. coli library and our Salmonella typhimurium library, so our two kind of main workhorse in the gram-negative libraries, and we exposed them to the three analogues of the drugs that we got sent of OPT200, and at 4 concentrations around the inhibitory concentration of the drugs. We also did an untreated control just to compare how all of these drugs act against just what's normally happening in the cell. So lots and lots of genes come up when you do this assay, and lots of genes have lots of different effects of different varying strengths. When we look at drug targets, we're looking at the biggest, most exciting hits. So at this locus, this is what the cell looks like normally. And when we add this OPT200 at different concentrations, we get loads of insertions upstream of this gene LPXD. So to cut a long story short, all of these insertions are driving increased expression of this LPXD. And this happens in E. coli, it happens in Salmonella, and it happens in the different analogues. So we're pretty confident that overexpression of this LPXD is causing a benefit to the cell, so it allows the cell to survive. And we can with talks with this Oplitec company, we decided that this drug was inhibiting LPXD activity. Therefore, if you overexpress it, you're outcompeting the drug. LPXD is an essential gene, so there is very few ways that would have been able to pick this up. And it just shows the strength of TraDIS-Xpress picking this up from all of the loci that are available. We tested this, we validated this, just by overexpressing LPXD on a plasmid vector, and we found 5 log-fold increase in the MIC. One thing that is important to point out as well, that obviously here, this is some of these kind of massive inserts are like a 14, 15 log-fold change from the control, so it's not exactly the same, but it's showing us that there is a massive effect. And then this proves with other research that Oplitec did that LPXD is this drug target. Now, as well as the drug target, like I said, there are loads of other genes that are affected by antibiotic exposure, and I've mapped them all out here in the, just to show that there is a massive effect on the cell. And one of the benefits as well of TraDIS-Xpress is that we can look for synergies. So when we exposed our same library to colistin, we found that all of these same pathways were hit. And when we exposed it to rifampicin, we saw that all these same pathways were hit. And when we use these two drugs together, we saw a massive synergistic effect that is outlined in that preprint if you want to have a better look. So this was just a flying talk to demonstrate all of this. So just to sum up, TraDIS-Xpress is ready and can be used for finding novel drug targets, finding novel drug synergies, and its application in the drug discovery pipeline will benefit the discovery of novel antibiotics for fighting AMR. Just want to thank everyone who's been involved in this project.